in this last lecture video for the interaction between cells and their environment, I'm going to be discussing on cell-to-cell adhesion and interaction. So this particular topic is a bit different from the previous video's topics because this is now interaction between cells. In the previous videos, we were talking about cells interacting with the ECM, or the extracellular matrix. So here we have an illustration of two types of cells, from the ectoderm and from the mesoderm, color-coded red and purple. And they are sort of mixed homogeneously in this particular scenario. After some time, we notice how the cells from the ectoderm, the one in red, actually aggregate themselves on the outer layers of this ball shape now, and the mesoderm cells are now confined in the center portion. So it's as if the cells know where they are supposed to be and with whom they are supposed to be. Mesoderm cells stay with mesoderm cells in the middle, and ectoderm cells stay with ectoderm cells on the periphery. After some time, they organize themselves even more. Here we can see the ectoderm cells flattening themselves up, and they're sort of in this uniform circular shape already surrounding the mesoderm cells. And the mesoderm cells, many of them have also organized themselves into these specially shaped cells that we see in the center. And later on, the mesoderm cells have organized themselves so much that there are now certain structures here in the middle of this cross-section. So here's an actual picture of heart cells and precartilage cells that were mixed together. So in this case, these are not strictly just ectoderm or mesoderm cells. They are from different organs or from different functions. Notice that when they're mixed together, the heart cells group themselves together on the periphery, while the precartilage cells also group themselves together in the center. So again, the question is, how do these cells know where to go and with whom to be with? So that will be one of the main topics in this discussion, cell-to-cell adhesion. So the cell-to-cell adhesion molecules include, number one, the selectins. So these are the molecules on the surface of cells that recognize carbohydrates. Okay, so you can see here, the ligand is actually an oligosaccharide, just like the oligosaccharide present on your red blood cells, indicating if you're either type A, B, AB, or O. So the selectins are the ones that recognize those carbohydrate ligands. Then here we have the immunoglobulin superfamily. So here in this first example, you can see here FN, or fibronectin, which is a part of the segment of your IGSF. And then in this portion, you have the immunoglobulin domain, which contain this portion, an RGD sequence. Okay, so RGD and RGD sequences are binding to each other. Then here we can see an immunoglobulin superfamily segment. Okay, and here this is actually the integrin from a leukocyte, a white blood cell that was sort of attracted to this IGSF segment. Here on the right, number 3, is our answer, the cadherins. The cadherins are calcium ions activated, as indicated here. So they need calcium ions. And they are able to stick with other cadherins of the same type, as if like a zipper. So look at this. This one sticks with the same similar subunit. And this portion also. So the longer the overlap, the stronger the adhesion between these two cells. And these two cells are similar cells, similar types of cells. Like your mesoderm cell, if this is a mesoderm cell, this one is also a mesoderm cell. And it's the adherence between those two mesoderm cells that allow them to recognize that they are the same. And therefore, they stick together. So take note, that's the cadherin, calcium ion activated. So in morphogenesis, in the formation of the embryo, we are familiar already that cells need to move to other locations, and they might need to aggregate together with other types of cells or with cells of their similar cells. So we see here how cells actually migrate from the epiblast. They go to the inner part of this embryo, okay, and then they stay here as mesenchymal cells, okay, as migrating mesenchymal cells. So what's the mechanism behind? They were once part of the surface cells, the epiblast, but then they have sort of decided to leave that and go inside. So take note of the concept of the cadherins again. Similar cells stick together because of the cadherins. These epiblasts have cadherins that allow them to recognize each other, and they stick together. There must be something happening to that cadherin that results to the epiblast to decide to leave the other epiblasts and go inside to become a mesenchymal cell. So the differences in the expression of the cadherins is the reason for this. So here we have a single layer of cells with similar cadherins, and so they recognize each other as similar, and they stay together in one layer. Here we see an illustration here in the center of these blue colored cells. These cells are starting to express a different type of cadherin. So there are different types of cadherins. And it's just restricted to the cells here in the center. So you see these cells now will recognize themselves as similar, while this cell recognizes itself as different from the cell that's next to it, because they're expressing different cadherins. So the result is they will try to separate from those other cells and will group themselves together into this neural tube. Okay? And so we now have accomplished the formation of a neural tube thanks to just the changes in the expressions of the cadherins in the cells. So those are the story of the cadherins and how they play a role in cell migration during the formation of the embryo in embryonic development. Now let's talk about intercellular junctions. Okay, so there are many types of boundaries between cells. All right, so here we have the adherence junction, which of course allows each cell to adhere to another cell, which has similar function with the desmosome, which is also for adherence. Then here we have tight junction. The tight junction's function is not merely to adhere cells to other cells, but it prevents or it locks the way from the outside here to the inside here. So it's sort of, there are staples in between the cells that prevent, say, extracellular fluid from going through this tight junction. Then we have gap junctions, whose functions are basically for communication between cells. So you can see here they're composed of sort of pores that connect the cells together. So let's first focus on the adherence junction and the desmosome. Okay, so you can see here how the cadherins and the cadherins are actually the ones that are involved in the recognition and in the adherence between two cells in the adherence junction. Okay? While in desmosomes, there's a special type of cadherin called desmocolin and desmoglycin, which also bind together to adhere the two cells together. So these are important differences between the adherence junction, the type of cadherins that are involved in the adherence, and between desmosomes. Okay, so we can see other structures here for the adherence junction. The cadherins are connected to catenins, and the catenins are connected to actins. Okay, so that's important. While in the desmosome, the desmoglycin and desmocolin are connected to desmoplakin, which are connected to intermediate filaments instead of actins. But the functions of these, although they differ in their structure, both adherents and desmosomes, is for the adherence between two cells. So here's an electron micrograph of a desmosome. So you can see here the plakin and then the intermediate filaments. And the two cells that are attached together, as shown in this boundary here. Next, the tight junctions. So the tight junctions are really, they appear like this, okay, three-dimensionally. And it's sort of like there's a staple wire between the two cytoplasmic membranes of the cells. Okay, and it's tight junction because this really doesn't allow for any fluids to pass through in between the two cells. So that's the meaning of tight in tight junctions. These proteins that serve as sort of staple wires are known as claudin, okay, because they cloud, perhaps they sort of cover, like clouding. And this is their detailed structure. So as shown here, in tight junctions, there will be a difficulty for extracellular fluid to pass in between cells because of the claudin. The next, the gap junctions. So the gap junctions are composed of proteins known as connexin, which aggregate together to form this rose-shaped channel we call the connexon. Okay, so the connexon is actually, it doesn't span in between cells, but it just stays in the membrane of whichever cell it belongs to. But connexons sort of act like magnets. They align themselves automatically once they are near each other. So the connexons will really align with each other, and the channel between them will also align, allowing for a passageway from one cell to another. So here we can see a micrograph of thousands perhaps of connexons that are aggregating together. So in a gap junction, the connexons group themselves together because, as I mentioned, they sort of act like magnets and they stay together to form the gap junction. Now, plant cells have a special type of this junction called plasmodesmata. Okay. So the plasmodesmata are the channels between the cell walls of two adjacent plant cells that allow them to communicate with each other. So here we can see a structure of a plasmodesma. Okay, we can see that there is actually a structure here that sort of inserts itself in the plasmodesma, and this structure is connected to the inner membranes like the Golgi apparatus and the endoplasmic reticulum of the plant cell. So substances are able to pass at the sides here of this structure. And the desmotubule, the green structure here, is the one in the middle. So there's also a study, a proof as to the presence of these plasmodesmata. Here we can see RNA that's being created by cells in the stele, okay, in the vascular tissue of the plant. And you can say how they are color-coded, and then later on, from just being confined in the stele, here shown in this small picture, they have now moved into the endothelial cells lining the stele. So how is that possible? There must be a passageway between the stele and the endodermal cells. And that passageway will be the plasmodesmata, okay, the gap junction. And lastly, we are going to discuss on a special type of extracellular matrix, the plant cell wall. Okay, there are two types of plant cell wall. The primary wall is the first wall to be created. Okay, and it's usually thinner, and it's for plant cells that have been newly formed. Okay, there's a primary cell wall, and in between two plant cells, there is a layer we call the middle lamella. So here's a structure of the plant cell wall, shown in a micrograph, and here's the drawing structure. So here we can see in the drawing, the main big structure would be this cellulose, okay, so take note, cellulose microfibril. And then, in parallel to that, filling in the gaps, would be the hemicellulose, shown here in purple. And we have glycoprotein, shown here in brown, and pectin, parallel to the cellulose microfibril, shown here in blue. So these are the structural components of a plant cell wall. So how is the cell wall formed? It's formed with the help of, first, the cellulose synthases, which are located on the membranes of plant cells. The cellulose synthases are shaped like these rosettes, okay? And they are sort of aligned together thanks to the microtubule, the cytoskeleton, that's hidden underneath the cell membrane. So they form a line. And as they form the cellulose microfibril, these are extruded out onto the outer layer of the cell membrane, and they form parallel lines of cellulose microfibrils. Okay? Again, the reason for this is underneath the cell membrane, there is this microtubule that controls the direction of the release of these microfibrils. Now, once the cellulose microfibrils are held in place, from the insides of the cell, from the Golgi apparatus, okay, the other components like hemicellulose, glycoprotein, and pectin are deposited from the inside onto the outside, thus forming a complete cell wall for the